Mr. Henderson. What do you expect to achieve with this project? What is the ultimate goal?
Yes, the ultimate goal is to achieve clinical qualification of novel biomarkers, innovative biomarkers, for either response to tumor therapies in a general sense or response to standard of care therapies. And at the same time to back this up with a more detailed examination of the properties, for example, of circulating tumor cells and circulating nucleic acids as a substitute or a surrogate for taking tissue biopsies from patients so that we have a less invasive diagnostic procedure.
Just to clarify a bit, could you perhaps explain with an example what would be the benefit of this?
I think the main benefit, there are two main benefits. One is from the point of view of routine clinical care, but also of drug development, that there is the possibility then of taking multiple samples from a patient during the course of treatment. So you start prior to treatment, then under treatment, and you can follow a more accurate time course. This is not possible if the only diagnostic method you have is to take a surgical biopsy. So with a less invasive method, then you have a more accurate time course of the way the tumor is responding to treatment. This can be of benefit, as I mentioned, in routine clinical care, but it is also a major benefit during the drug development process, especially today where we have more and more targeted therapies coming into development. Targeted therapies are generally very specific and have far fewer side effects than classical cytotoxic oncology drugs. And this means that one of the major problems we have in drug development is to decide when the dose is correct. And so dose finding is something that would benefit enormously from biomarkers of this kind.
Okay, thank you. Now, what would, in very concrete terms, be the contribution of the industry in this project?
I think there are a number of ways that industry can contribute, and a lot will depend, of course, on the exact program that comes out of the consortium that's selected. But obviously the pharmaceutical companies have a lot of experience in design and conduct of clinical trials. And we also have considerable experience in analyzing samples from clinical trials. From a practical point of view, developing analytical methods and also qualifying analytical methods for clinical use. And of course, if it's necessary, we have collections of data on characterization of tumors so that we know something about the distribution of biomarkers in particular tumors, in particular indications. This can also be a great help for the consortium members.
Okay, thank you. Now, why would small companies or academic teams consider to take part in this project? What very concretely is in it for them?
I think a great deal. I think one of the features of working in a consortium of this kind is the idea that together we are much stronger than when we're standing alone. And one of the basic ideas of working in this way, I think, is to create synergy among the members. And this means that academic laboratories may be able to more easily to form partnerships with other labs, and also, of course, with members of the pharmaceutical industry, which otherwise might not be quite so easy to put in place. And then from the point of view of an SME, this is an opportunity, for example, if they have an innovative platform for assessing biomarkers, then this would be a platform where they would be able to bring this relatively rapidly into a setting where it's possible to do some form of validation study. And so I think those are the areas where the consortium can really help.
Okay, thank you very much.